All right, recording begins. It is October 17th, 2025. This is Biology 105, and we're talking photosynthesis. So first thing I wanted to bring up, right, is that your data from the photosynthesis lab should be, some of your lab data is already posted. Okay, if you want to graph your own bench's data, it's cool to do. You want to see, what results you got, So you can see these two files right here, Thursday 2 p.m., Thursday 5 p.m., you already have data uploaded. If your data doesn't get uploaded by the time you're ready to do your assignment, your pigment graphing, pigment extraction and identification lab assignment, there is a backup data set that you are welcome to use. But if your data is available when you go to do the assignment, it'd be fun to do your data, do your own data. I've got a tutorial video on how to do the figure. It's right there in the module for week 8. And are there any questions about that? Yes?
Once we get back the data, how will we get back our data that we put into the machine?
Oh, it shows up like this. It'll be in these Excel files. It looks like.
Each group?
Yeah. So you'll check this out. Each bench has all these different wavelengths and then the different pigments.
I'm guessing bench A is the one at the front.
I'd have to assume so. Yeah. I would have to assume so. And then graphing this data is really friendly because it's already all set up in columns for you. And I'm really trying to get you comfortable with Excel. I'm not so worried about you knowing how to format the data correctly. But if you want a pro strategy, always put things into columns in Excel. It loves columns. But you can see right here, like it's pretty quick to do this. I'm doing it right now. So there's no need to put this off. Just go ahead and do it, follow my video tutorial. And look, I've already got an absorbance spectrum done, just like that. I just have to add an axis title to axis titles, right? It is very quick, wavelength and absorbance. So hey, bench A on Thursday at 2 p.m., nice job. This is what we expect an absorbance spectrum to look like. I wanted to spit one of these out because we're going to look at some idealized absorbance spectra in our photosynthesis PowerPoint. So that's that. Also, it's Friday. That means that your lab midterm, your online lab midterm is due by 11:59 p.m. tonight. So, many people have done it. Well done. If you haven't done it yet, do it today. Cool? Any other questions? Bless you. Any other questions? All right, solid. Then let's just get back into the photosynthesis PowerPoint. Where's my absorbance spectra? Here they are. Okay, so we talked about chloroplasts. We talked about chlorophyll. Remember, the chloroplast is the organelle. Chlorophyll is one of the pigments. Chlorophyll is the primary photosynthetic pigment, by which I mean the pigment that absorbs light where the energy is going to be directed into photosynthesis. There are all sorts of other pigments that may or may not be involved directly in photosynthesis. The chlorophylls are the primary ones that actually drive photosynthesis. All right, look at this figure right here. There's actually 3 figures. Let's look at all of them. This is old data from a previous lab back in like 2019. They generated this figure. It graphs the absorbance of a bunch of different pigments across a broad spectrum of wavelengths of light. And it indicates, oh, these wavelengths here and these wavelengths here are absorbed by chlorophylls, okay? Check it out right here. This is the absorption If you look on this figure, we've got the action spectrum, and this action spectrum is meant to represent how much photosynthesis gets driven by each wavelength of light. So in the equation for photosynthesis, one of the outputs is oxygen. And in this figure, they're measuring photosynthetic rate by quantifying the amount of oxygen being released when you blast a plant or an algae with a specific wavelength of light. They show that these wavelengths of light are the best at driving photosynthesis, along with these wavelengths of light, but these wavelengths of light are not very effective at driving photosynthesis. And why? Because the wavelengths of light which are absorbed by the photosynthetic pigments are those that can drive photosynthesis. Reasonable enough? Plants appear green because they don't absorb green wavelengths of light. Or they absorb less of the green wavelengths of light. And that's it. Now let's get into the process, okay? Photosynthesis has two separate sets of reactions that we're going to look at, all right? Remember in cellular respiration, there were 3 different stages. Well, in photosynthesis, we're going to look at 2 sets of reactions, each one of which has its own stages. So it's going to get a little bit complicated, but we're going to be focusing on the light reactions primarily, and the Calvin cycle will be mainly just, we'll hand wave past it. All right, so here we go. Let's get our overview. The light reactions. The light reactions use light energy, and they generate ATP and NADPH. These are going to carry that energy into the Calvin cycle. All right? So NADPH is like NADH, but it has a phosphate group on it. It serves exactly the same function, but this is the electron carrier in photosynthesis. A little mnemonic device, P for plant, or P for photosynthesis, as opposed to NADH in cellular respiration. I'm going to pick up my energy a little bit. I do not need to be putting you all to sleep on a Friday in week 8. Bah! I know, this is very scary. We're all energized now. Let me have some of my mysterious fluid. Here we go. Oh, it's so good. Oh, it's so great. Okay, the light reactions. Light energy is converted into chemical energy in the form of ATP and NADPH, right? You know what ATP is. You love ATP. You're making it and burning it right now. Keep up the great work. NADPH has reducing power. It's going to carry some electrons that are in a high energy state to the Calvin cycle. And all you need to know about the Calvin cycle is this. Ready? It's going to take the energy from ATP and the electrons from NADPH and use them to build sugar. That's it, all right? You're taking a very low energy starting product of carbon dioxide, and you are jamming energy into it, and you are attaching electrons onto it in an endergonic anabolic process to build up a high energy organic molecule. The energy for building the sugar comes from ATP and NADPH. The ATP and NADPH were generated from the light reactions. The energy to drive the light reactions came from the sun. Cool? The electrons that are carried by NADPH, they have their origins on the water molecule. And if you remember, the electrons on a water molecule are at an incredibly low energy state. So they got to get jazzed up to a high energy state, and the process of photosynthesis will do this by using light energy. Ready? Let's get into the details. Let's take it to the bridge. Here we go. NADH, NADPH. There's a P. All right? Overall, functions the same for our purposes. But NADPH is in photosynthesis, and NADH is in cellular respiration. So rest assured, there's going to be a midterm question where it's like, what's carrying the electrons in photosynthesis? You know? And it'll be, NADPH for photosynthesis. The P is for plant. It's not really, but that's fine. Okay, so we're going to zoom into the light reactions. That's what we're going to be focusing on. I know I'm getting a little repetitive, but I really want to start on some firm footing here. We're going to zoom into the light reactions and focus a lot here. And you're going to see a ton of similarities between the light reactions and cellular respiration. I'm talking electron transport chain. I'm talking dang old ATP synthase with a proton motive force, all that stuff. Here we go. Let's do it. So in the thylakoid membrane, right, the innermost membrane of the chloroplasts, you've got these protein pigment complexes called photosystems. Hey, you've got these pigment protein complexes called photosystems. And these are the light harvesting component of the thylakoid membrane. So it's a bunch of pigments embedded in protein, and that protein pigment complex is embedded in the thylakoid membrane. Any questions thus far? Okay, let's keep on going. What does this photosystem do? It takes a low energy electron and blasts it to a high energy state. That's what this is designed to do, okay? And then it can, so check this out, right? You've got this special pair of chlorophyll molecules right here. You focus all of the energy from these pigments into this special pair of chlorophyll molecules, all right, which has a special name, which we will talk about in another slide. You blast an electron off of this with all that energy, and now it really wants another electron. It was holding on to that electron very tightly. So it's going to take that electron from a low energy state source. It could be water or from just a very electronegative electron carrier. Anyways, don't worry, we'll see that a little bit more. We will get into this, how the electrons go from photosystem 2 to photosystem 1 in just a moment. So this is probably more background than you necessarily need. Here's what one actually looks like in more of an accurate model. Like, look at all these pigments and the proteins and all the alpha helices and all the structure. So of course, nothing's as nice and simple as the diagram. But there you are, right there. Okay, cool. So the photosystem and all of the pigments embedded in the photosystem. So you got all these pigment molecules. They get hit, they're all getting hit by energy, and they're all sort of acting like antennae. It's more like they're acting like a, oh my gosh. You all drive to Vegas from here regularly, right? I lived in Riverside, and that was one of the few things you could do, was you get to Vegas in like an hour and a half. This is horrible. None of these expos work. Let's just bust out the notebook, which means I walk over here for a pen. And I walk back over here, you see. And then I hit the button. Smooth. That went so smoothly, just like I planned it. All right. You know when you go out to Vegas and you see the big field with all the different mirrors that are like blinding? You know what I'm talking about. The solar farm, exactly. They do the solar farming like this, where the sun hits and it all gets blasted and focused onto this tower in the center of all of these mirrors, right? And then they actually can harvest this. Sometimes it's by they actually boil some fluid in here that spins a turbine. There's different, methods of actually harvesting the light. But you focus all that energy into a single point. And that is what's happening with all of the pigment molecules in the photosystem. They're all getting hit by light, they're all vibrating, and they do energy transfers, go boom, 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 and they force everything into this special reaction center chlorophyll complex. So the reaction center is the portion of the photosystem that's actually going to allow an electron to get kicked up and transferred to another molecule. Okay? My point here is just that there's not one pigment that absorbs the light. These diagrams can trick you into thinking like, okay, the light hits a single pigment, and then that pigment transfers energy through a bunch of different pigments to hit the reaction center eventually. It's more like all of these are getting hit by energy, and they're all focusing their energy into this special pair of chlorophyll molecules right here. That energy is used to kick an electron from a low energy state on this pair of chlorophyll molecules to a high energy state, where it then gets moved on to this thing called the primary electron acceptor. And now it's at a high energy state, and it can go boop, 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 and fall down an electron transport chain and pump protons, just like in cellular respiration. So what's the difference here? The difference is when a heterotroph eats something, all right, and breaks it down via cellular respiration, the electrons were already in a high energy state. And respiration lets them fall from that high to a low energy state. In photosynthesis, the electron first must be elevated to a high energy state by solar radiation. Okay, cool? So in linear electron flow, this whole process right here, there are two photosystems involved, photosystem 2 and photosystem 1. They are named in order of discovery, not in order of operation. So photosystem 2 operates first, and then photosystem 1 operates second in this process, which is frustrating, but it's because this one was discovered first, and it's also most likely that photosystem 1 evolved first, but we'll get into that point in a little bit. Alrighty. So this is called linear electron flow, or the Z scheme of photosynthesis, because if you turn your head on its side, you can see how this is a Z shape. And what is that Z representing? Energy level of the electron, okay? The electron starts at a low energy state here in photosystem 2. It gets blasted up to a high energy state. It falls down an electron transport chain to a low energy state in photosystem 1, and then it gets blasted up again to a high energy state in photosystem 1. How are we doing so far? Question, and then I'll get you next. Get you next.
We want to go from high to low. Wait, I'm already getting confused.
No problem. This is super confusing. So check this out. Both the light reactions of photosynthesis and cellular respiration have the goal of generating ATP. Both of them carry out chemiosmosis. In photosynthesis, you have to get the electron to a high energy state first. So that's this process. Once it's at a high energy state, you just boop, 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 drop it down the electron transport chain, and that is going to generate ATP via oxidative phosphorylation, via chemiosmosis, right?
Okay, but it's cellular respiration.
It's basically the same process.
Same thing. Okay.
Yep, and yeah.
So for photosynthesis, the ATP isn't produced until the electron transport chain.
In both of them, you need the electron transport chain to pump the protons to generate the proton motive force. Let me find another image that I think will be helpful. Okay, this is way more similar to the image that you've seen already in cellular respiration. Check it out. An electron goes to a high energy state. In respiration, it was already at a high energy state. and then it falls down an electron transport chain. As it falls to a low energy state, that energy is used to pump protons, generating a proton gradient called the proton motive force. And then those protons are forced to flow through ATP synthase in order to generate ATP. So this portion here, the electron transport chain and the action of ATP synthase, is, for our purposes, identical between respiration and photosynthesis. The only difference is, did the electron start at a high energy state, or did the electron get elevated to a high energy state?
That's what I was confused about.
Did that, okay.
Photosynthesis, it has to get to that high energy state, but cellular respiration is already there.
Yes, absolutely.
That's what I'm confused about. Thank you.
And let me just add one more last complicating factor to it, right? So check this out, right? In cellular respiration, once the electron falls to the low energy state, you're done with it. You put it on oxygen and it becomes water, right? In photosynthesis, that electron falls to its low energy state, and then you elevate it to a high energy state again, because you need to generate, if you remember, NADPH. All right? In cellular respiration, you consume NADH. That's got the electrons that get fed in here. But in this process, you got to generate NADPH at the very end, because you need both NADPH and ATP to drive the Calvin cycle. You need both of them. So the light reaction must produce both NADPH and ATP. All right? It generates the ATP via oxidative phosphorylation, just like cellular respiration, and it generates the NADPH via the activity of photosystem 1, where an electron gets elevated to a high energy state, and then instead of going down in the electron transport chain, it just gets slapped onto NAD+, and you get a charged up electron carrier. A question, yes? You know what? That's absolutely fine. Let's go ahead and dial back, and maybe this will spark your memory. So now that we've looked at this whole figure here, right? And this is one of those like big money figures where it's got everything you need to know about the light reactions in it, but just looking at the figure doesn't mean very much without the discussion of all the steps. So let's go ahead and talk about well, we're going to be focusing a lot on the activity of photosystem 2 and photosystem 1 and the Z scheme of electron flow, or linear electron flow, where electrons go from water through this whole system and end up on NADPH. Cool? Awesome. The photosystems. Photosystem 2 functions first. All right? The reaction center special pair of chlorophyll molecules is called P680 because the wavelength of light because light with a wavelength of 680 nanometers, or radiation at the wavelength of 680 nanometers, is the best at driving it. It absorbs that wavelength the best. It's the best at charging it up, okay? So when P680 loses an electron, remember, it's just a special pair of chlorophyll molecules, but when it loses an electron, it is called P680 plus, because it's lost a unit of negative charge, so now it has 1 unit of positive charge. Reasonable enough? P680 plus is the strongest known biological oxidizing agent. What does that mean? It means that it is very good at pulling electrons off of other things. It's incredibly electronegative. It's an oxidizing agent, so it's going to oxidize something else. Now, can anybody tell me what P680 plus is going to oxidize?
Let me guess, water?
Yes, 100%. 3.5 points. Because you said, let me guess. So, you know, the confidence would have brought you up to 4. Yeah.
I know water is very, it holds onto them really tightly, so it would need a very strong electron puller.
That just earned you the other half point. Now you're at 4. That's solid. That's exactly it. And everybody catch that right? oxygen doesn't want to give up electrons. Water doesn't want to give up electrons. So in order to break water down and pull electrons off of it, you're going to need a very strong oxidizing agent, like P680+. So this right here is going to be where we consume the water and where we generate the oxygen. This step right here is where all the oxygen comes from in photosynthesis. All right, great. Photosystem 1, the other photosystem. It's got a special pair of chlorophyll molecules in its reaction center, and it's called P700. It's called P700 because radiation at 700 nanometers is the best at exciting it. It absorbs that wavelength of light the best. All right? So after the electron transport chain, photosystem 1 acts. And what photosystem 1 does is it blasts an electron to a high energy state, and then it puts that electron, there's an intermediate step with a ferredoxin thing, don't worry about it, okay? But it pops it on to NADP plus to make NADPH, all right? So this step is all about generating reduced electron carriers. Photosystem 2 generated high energy electrons so they could fall down an electron transport chain to generate ATP. But photosystem 1 generates high energy electrons and then just puts them on to NADP plus to make NADPH. And now that's another form of energy, right? A nice reduced electron carrier. Cool? All right, awesome. Good. Yeah, P700 plus does not take an electron off of water. It takes an electron from the end of the electron transport chain. And if you remember our electron transport chain talk, at the end of the electron transport chain, the electron's at a very low energy state, right? So it's not very reactive. So you're going to need a powerful oxidizing agent to accept that as well. Cool. Okay. The steps of a linear the steps of linear electron flow. Here we go. Photon blasts photosystem 2. All this energy goes off. Eventually, the energy gets funneled into P680, and the electron from P680 gets kicked onto, just a nice, convenient term, the primary acceptor. All right? So remember primary acceptor, terminal electron acceptor. All right? So the primary acceptor is going to take that electron. Now, in order to regenerate P680, you're going to take electrons off of water. So P680 plus is going to strip electrons off of water. That's going to reduce it back to P680. The hydrogen ions, the protons, get released into the thylakoid space. Okay? This is beneficial, okay, to know, because you might be asked questions about which direction the protons get pumped, okay? And if the protons are released into the thylakoid space already, it makes sense that that's where you're concentrating protons, right? There's a highly evolved system, so it's going to have efficiencies like that built into it, usually. So this is all about concentrating protons in the thylakoid space. Anyway, breaking down water releases a proton, releases a couple protons, and releases some oxygen. Cool, this is where the oxygen comes from. The electron's now at a high energy state, and P680 has been regenerated, so this can happen again. Now, the electron can fall down an electron transport chain. There are, of course, many more players than just the plastoquinone, the plastocyanin, the cytochrome complex, all this. The electron transport chain is complex, obviously, right? You know this. It's falling from a high to a low, doop, 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 energy state, pumping protons, making a proton motive force, carrying out chemiosmosis, giving you ATP. Cool? Now the electron's at a low energy state, and that electron is going to be taken up by P700 plus. Okay? So that electron's at the end of the electron transport chain. P700 becomes excited by light energy and blasts off an electron to a primary acceptor. It's the same basic system as in photosystem 2, except instead of taking electrons off of water, this is taking electrons off of plastocyanin, or the end of the electron transport chain. Okay, I don't want to call it the terminal electron acceptor because really P700 plus is the terminal electron acceptor here. And then it is going to drive the electron back up. The terminology doesn't really fit. So just forget I said anything, okay? And then the electron that is high energy here is going to I need to skip ahead a slide, is going to just fall down a very, very short electron transport chain. It's not an electron transport chain for pumping protons. It's just an electron being transported to NADP plus to make NADPH. All right? So ATP here, NADPH here. Cool? All right. Please.
So this is sort of not maybe that relevant. How is energy passing from the individual pigments to the central reaction process?
Yeah. Oh, totally, yeah. So it's like an electron goes up, you hit the reaction, not the reaction center, you hit the antenna complex of the chlorophyll molecule. It's got that magnesium in the center. It's got these nitrogens there. The electrons are all in weird states of movement because of the differences in electronegativity. And when the radiation hits the center of that, the electrons go up to a high energy state, right? And then the electrons fall down to a lower energy state, and they re-radiate energy. So as the electrons go high to low energy, that energy gets released. And it's not really like heat. They release some heat, but they also radiate literal electromagnetic radiation, which then is picked up by other pigments in close proximity to it. They're all just close to one another, so they're all going high energy to low energy, and then another one goes high energy and then to low energy. Yeah, 100%. They're all doing the wave, back towards like the back of the room, and then somebody in the back of the room is getting all those positive vibes, all that energy, and they're gonna just blast an electron off, by which like they throw their hat in the air. That's a perfect analogy. Yeah, solid. And as with all of this stuff, there's more to it, but that's a pretty good understanding of it, honestly. It's all about absorbing energy and then re-radiating energy. Yeah, I love it. A question, please.
Yes. So does photosystem 2 produce the ATP while photosystem 1 produces the NADPH?
That would usually. Here's the problem. This process here is linear or non-cyclic electron flow, and it's sort of the idealized version of photosynthesis. But also there's a phenomenon called cyclic electron flow, and in cyclic electron flow, photosystem 1 takes those electrons and feeds them into this electron transport chain to generate ATP.
So photosystem 1's a little bit more of a cycle, while photosystem 2 is very linear.
In cyclic electron flow, photosystem 1 is the only thing that can carry out cyclic electron flow. So photosystem 1 is capable of carrying out a cyclical process. Photosystem 2 always carries out the linear process. Yeah, that's what you said. I just rephrased it back at you. But yeah, solid? I love it. Okay, but we're focused right now a little bit on non-cyclic electron flow, which is going to generate ATP and NADPH. Yes, please.
Would you have to memorize all the steps?
Oh, no. No. For anybody listening to the recording, the question was, do I want you to memorize all the steps? No. But I want you to memorize what photosystem 2 does, what photosystem 1 does, where does the oxygen come from, where does the ATP come from, cyclic versus non-cyclic electron flow, which thing carries it out, where does the NADPH come from? So memorizing each one of the steps is one way to accomplish that, but the way that they're laid out here as like step 1, step 2, step 3, step 4, it doesn't work for my memorization. For me, it's better to look at something like this, in all honesty. Like, this is what I want you to know on this figure right here, all right? It lays out all the steps, but you don't have to memorize it as step 1 is this, step 2 is this. I wouldn't ask you a question like, what is step 3 of linear electron flow, for example? That makes sense? Awesome. Yeah, no worries. Okay, cool, good. So non-cyclic electron flow, I've talked about it, I've explained it. I think I'm going to say I did well enough. All right. Cyclic electron flow is this process here, where photosystem 1 just feeds electrons back into the electron transport chain that generates ATP. There's different theories as to why this process occurs. It's very likely that it's an evolutionary holdover, because most likely photosystem 1 evolved first because it can carry out production of ATP and NADPH. But then having a second photosystem probably increased the efficiency of this, so you don't have to constantly alternate between, well, now I'm making ATP, but now I'm making NADPH, and now I'm making ATP, and now I'm making NADPH. Linear electron flow generates ATP and NADPH in equal amounts. A question?
Other classes where we'll go over the variations in photosynthesis with like C3, C4, and CAM.
Oh, we're going to talk about that here. Yeah. At the end of the PowerPoint. Yeah. Oh, but sorry, did I cut off your question? Oh, good. Yeah, totally. We're going to talk about that. It's super important. Yeah, question.
So just to clarify, cyclical electron flow, photosystem 1 makes ATP and NADPH.
Yes. Well, I'm sorry, actually, in cyclic electron flow, which is specifically this, no NADPH is produced. It's only ATP.
Okay.
Yeah, but yeah,
NADPH is produced, just ATP.
just
And it
ATP.
keeps going and going and going.
Exactly.
And then in linear flow, it goes from photosystem 1, ATP, photosystem 2, NADPH.
Photosystem 2, ATP, photosystem 1, NADPH. Because they operate in they count down.
Getting used to it.
Totally. It's super tricky. And yeah, if this is your first time getting like a whole discussion of photosynthesis, this stuff is so complicated. It does not, your brain isn't just gonna absorb this all perfectly. It's gonna take some review time, I guarantee it. Yes?
What would you say is more complicated, photosynthesis or cellular respiration?
Photosynthesis. Yeah. I mean, it all depends on if you're having to memorize these cycles. But the Calvin cycle and the Krebs cycle, they're the same. They're the same level of. I think they're just as complicated as one another. Photosynthesis has this additional process of the light reactions on top of it. Yeah. But that's why I do it respiration first, then photosynthesis, because I feel like if we understand respiration, the photosynthesis is a little more approachable. Because this whole step right here, I don't even have to explain it. We already know what it is. It's just oxidative phosphorylation. Okay, we've talked about non-cyclic electron flow. We've talked a little bit about cyclic electron flow. Here's another couple of diagrams. Like I mentioned before, this is a big important one. We've been focusing on the light reactions, where the oxygen comes from, and how they generate ATP and NADPH. Questions? Okay, I'm gonna go back over here. Here's a diagram of this. I don't know, this might be useful, okay? Check it out. These folks have big hammers providing energy to elevate electrons to high energy states. Then they throw that electron down a ramp to a lower energy state, and that drives a little spinning wheel, and that spinning wheel makes ATP. All right, it's ATP synthase, you see? And then you got this next lever here being hit by a photon, elevate it to a high energy state, and then leave it at a high energy state by throwing it into a bucket. Does that helpful? I don't think so, especially, but then again, I've had to learn this process so many times that to me it's kind of like, well, this is obvious. Like, you can see how this is a bad analogy because the wheel is like ATP synthase, but the electrons never flow through ATP synthase. So there's a problem right there in it. But the key thing here, why this can be helpful for folks, is it goes from a high to a low to a high energy state in the light reactions, and it stays at a high energy state right there in that NADPH bucket. Cool? Cool. Let's keep on keeping on. Here we go to the next thing. No oxygen released in cyclic electron flow, probably evolved before non-cyclic electron flow, because hey, if you're an organism, maybe you're just using this to generate ATP, and that's how you survive, okay? Anyway. Oh yeah, some people say, like, why does it still happen? And people will say, oh, it's because if you have too much light, if the light's too intense and you don't have enough NADP plus to lose these electrons, you don't want to leave everything all charged up, so you can just shove things back through the system. But evidence is contradictory on that, you know. Anyway, cyclic electron flow. Here's those stages. Look, this is just this figure broken up into a bunch of different pieces. So we could talk about, like, look, here it is again. The electron goes to high energy. The electron falls to low energy. It pumps protons. The electron goes to high energy. The electron goes to NADPH. The proton motive force is harvested by ATP synthase to generate ATP. And then both ATP and NADPH go into the Calvin cycle. Cool? All right, sick, thank you. I appreciate the thumb, it's always useful. Calvin cycle time. Are you excited about it? You should be, because you're not gonna have to memorize it, which is very nice. So there's all of these steps of the Calvin cycle, and I will be specific and honest with you about what you need to memorize in order for the learning objectives of Biology 105, okay? The main thing, this bullet point right here, is the best sort of summary of the Calvin cycle. Check it out. You're making glucose, right? What do you need to make glucose? You need carbon. That carbon comes from carbon dioxide. You need electrons, all right, to build up that new structure. Those electrons come from NADPH. And you need energy because you are taking something small and making something big in an anabolic reaction. It's endergonic. And so to do that, you gotta have energy, all right? And the energy comes from ATP. Now, it's always taught in Bio 105 and even in, some like botany courses and stuff, that this process makes glucose, but that's not actually super accurate. The Calvin cycle generates this molecule called G3P, which is this, it's got a phosphate in it, all right, so it has some energy, but it's this intermediate molecule, which then can go on to be used to build all sorts of stuff. So photosynthesis doesn't really just make glucose. Actually, it makes a precursor molecule called G3P that can be used to make a bunch of other organic molecules as well. Now, I say that, but for the learning objectives of a Biology 105 course, the Calvin cycle takes carbon, electrons, energy, and makes glucose. All right, that's like, your takeaway learning objective that you have to know. Okay. Good. Sometimes the light reactions and sometimes the Calvin cycle is called the dark reactions because the Calvin cycle doesn't actually need light in order to occur. The energy for the Calvin cycle comes from ATP, right? But that ATP came from when it was light. So calling them light reactions and dark reactions, we've kind of moved away from that terminology, and now we call it the Calvin cycle instead of the dark reactions. Anyways, let's look at how it actually operates. This is a video here that you can watch when you want to. Whoa, look at all those things spinning around. Look at those G3Ps. Look at that stomata. Look at all the oxygen coming out of it. Whoa, flying around. Very cool. You can watch the video. I'm going to give you the lecture. The video is embedded in the PowerPoint. How are we doing on time? 2 minutes. That's okay. There's only 16 slides left. Let's just absorb this. Go. All right, cool. We have fun. We have too much fun here. Here's the Calvin cycle. All right, a lot of these slides that are coming up are all just review slides, same slides you've seen before. So let's just look at the Calvin cycle real quick here. And I'll tell you what you need to know about it. As always, this is a gross oversimplification of the actual pathway. There are things coming into here and leaving this process. It's not perfectly cyclical, all this stuff, okay? But here's what I want you to be aware of. There is a very, very important enzyme here called ribisco. It is, by some estimates, the most abundant protein on the entire planet, okay? The details of the structure of ribisco have killed more people due to starvation than war, all right? This enzyme is the most important enzyme for global stability of bioenergetics, all right? This thing, this little purple box right here. So I'm not gonna spend a lot of time talking to you about like, what's the structure of these different molecules? Here's what happens. There's an enzyme called ribisco. It takes CO2, all right? It attaches it to this ribulose bisphosphate molecule, okay? That's what it does. From a 5-carbon molecule, it makes a 6-carbon molecule, which then immediately splits into 2 3-carbon molecules. This is carbon fixation. So when people talk about carbon fixation for the perspective of the carbon cycle, they're talking about this. CO2 in the atmosphere gets put into stable organic molecules. And then this cycle is just going to run and shoot out G3P, which is going to go and be used to make other organic molecules. It's going to keep running. It's going to make ribulose bisphosphate again, and it's just going to do it again and again and again. This process requires energy. This process requires some electrons. Those are provided by the light reactions. That's all the Calvin cycle stuff you need to know, okay? That's really it. We're going to focus a lot on this carbon fixation step because, as I've hopefully laid out for you, the activity of ribisco is incredibly important. But you do not need to memorize any of these. This you've already seen. This you've already seen. You've seen it. You've seen it. You've seen it. Ribisco. This is where we will come back to on Monday, and then we'll actually be able to get into our week 9 material in week 9. It's crazy for us. Have a great day, everyone. Have a wonderful weekend. It's like weekend time. Farewell. Let me stop the recording before I answer any questions here.